Okay, so walk us through the challenge here of, let's say, even just like solving this problem. So you have a sequence, a sequence of amino acids. That's how do you even
Correct.
represent the protein? And okay, so
Correct. Yeah.
a
Yeah.
protein is just a bunch of amino acids,
Right.
um, and they're organized in some kind of bizarre shape or form or something like that. But really, it's just a list.
It's exactly. It's a list. It's a sequence. The way it's given to the computer. Now, what gives a function to a protein is actually a combination of one, mainly its overall 3D structure, um, and which part moves in the structure, which parts are rigid,
Yeah.
and two, the biochemical characteristics of each amino acid, plus where they are.
Mm.
So, for
Okay.
example, if you have some amino acids that are hydrophobic, right? They repel water. If they are outside, if with the final 3D structure they are outside, the whole protein is going to be hydrophobic. But if somehow they're embedded, and hydrophilic protein are outside, then the protein is going to be hydrophilic. So it's a combination of a lot of factors
Yeah.
that end up to what the function is going to be. Um, and hydrophobic.
So when you look at a data set, like at a database like this, if we were to download this, each row would be what? Just like a sequence of amino acids, like, and how there are 20 amino acids, right? How many amino
There are,
acids?
yeah, 20 amino acids.
So it would
And
just be acid 1, acid 5, then acid 9, then acid 1 again, acid, right? It would
so the
just,
vocabulary is 20. Yeah, the vocabulary is 20 amino acids, and the sequence length can be anywhere between 20 and 2000 or even more.
yeah.
Uh, so you have it's like every protein is a sentence basically, and you have a corpus of text, and you want to classify the sentences. Let's say, yeah.
Now, in this case, we if the only thing we had is is just, let's say, like the alphabet, right? So we just like that sentence. That still doesn't tell us anything about the shape of it, right?
Exactly.
So okay, so let's imagine you have that sequence, and what you're trying to classify for this model is the destination, right? The
Right.
localized kind of where it's
Correct.
going to be. Okay, so before there was machine learning applied to this, are there some trivial ways by which you could just sort of by eye look at the sequence and say, oh yeah, this is going to end up here and there?
Yeah. So given a data set, what people would do mainly is look at sequence identity. So given some sequences that you already know go in some places,
Yeah.
if you get a new sequence, what you could do is running this new sequence against all known sequence and see how similar it is.
Mm.
And if it's very similar to something we know end up in the nucleus, for example, you could hypothesis. You can make the hypothesis that it would also end up in the nucleus. This approach has a lot of caveats, where machine learning and specifically language models, can come into play here. But that's, I think, again, I'm not a good canonical bioinformatician, but that's
Yeah.
definitely what people used to do.